All right, let's continue with Chapter 7 on quantitative genetics, explaining the evolution of complex traits, complex phenotypes. So picking up where we left off, this section from our book starts off by pointing out that those traits that are influenced by many genes, human height, for example, has polygenic inheritance, that to understand the evolution of these kinds of traits requires new tools. That's what we've been seeing in this chapter so far, and here comes another one. So what's talked about here is how the mechanics of meiosis can end up linking genes on chromosomes, alleles on chromosomes, such that they get passed on as a package, generation after generation. And these packages of genes that are close together on chromosomes might have a benefit in being so close together and being inherited as a package, might be involved with complex adaptation. So let's start off then by quickly, mentioning a couple things about meiosis, in particular, independent assortment, something we reviewed a few chapters ago in going over some genetics. So if you remember from independent assortment, homologous chromosomes lining up on opposite sides of a metaphase plate, such that there's an equal chance in any cell that they'd be on one side or the other. And so when those chromosomes segregate and go into different gametes, the chances of two loci on different chromosomes landing in the same gamete is 50%. It's a 50-50 chance. It all boils down to independent assortment. Now this phenomenon, where there's literally a 50% chance of two loci on different chromosomes ending up in the same gamete, that's called linkage equilibrium. Linkage equilibrium occurs by definition, under that circumstance. Now, the degree to which alleles are inherited on the same chromosome, not different ones, but the same one, is called linkage disequilibrium. So the closer that two genes are on the same chromosome, putting them closer, then the stronger that linkage disequilibrium is, the more that they're going to be passed on as a package to the next generation. Because they are so close together, or when genes are very close together, crossing over, when homologs crossing over is less likely to break up those alleles that are really close together and put them on different chromosomes. So that's the effect of linkage disequilibrium. The closer that those alleles are on the same chromosome, the tighter the packages that they're going to be, where crossing over does not separate them and put them on different chromosomes. In the figure, it's a big giant figure from your textbook, copied and pasted on here. In part A, the upper left, it's showing physical linkage of alleles at multiple loci. Okay, so on the top of figure A, you see two homologous chromosomes. One is kind of an orangish color, one's a blue. And then there's 1, 2, 3, 4 pairs of homologs below that, just showing them as like different sizes. So on that first pair of homologs, you can see that they're depicting multiple loci on the same chromosome. And those multiple loci on the same chromosome, by definition, those would be in linkage disequilibrium. But the farther apart that those loci are from one another, then the less linked that they are. If you look at that top chromosome on the far right, you see two loci that have been placed really close together. Those are tightly, physically linked. So there's a strong linkage disequilibrium between those. Now look at its homolog, the blue chromosome right below it. There's a locus, a different locus, and that one compared to a locus down on the bottom Okay, the chromosome on the very bottom, well, those are on different chromosomes. They're not physically linked, so that would lead to linkage equilibrium. Okay, so that whole figure is intended to help you understand linkage, equilibrium, and disequilibrium. Now let's look in figure B, because it's showing you how over time, over generations, linkage disequilibrium, the region of linkage disequilibrium gets smaller and smaller and smaller because of crossing over. So on the very top of the figure, it's showing you, okay, there's a chromosome that's got a gene on it, for example, a gene that can increase the chances of breast cancer. And so there's a mutation in that gene, the BRCA1 gene. So you get that mutation now can increase chances of breast cancer. Now on that same chromosome, really far down from it, you can see in the figure, there are single nucleotide polymorphisms, SNMPs, and those can serve as genetic markers, like little beacons on the chromosome. And what the figure shows is that over multiple crossing over events, okay, in subsequent cells, based on where the chromosomes, the homologs, are overlapping one another and crossing over is happening, the region of that disequilibrium can get tighter and tighter and tighter and tighter, smaller and smaller and smaller. And so that's showing you that the degree of linkage disequilibrium on a chromosome erodes over time. So with this in mind, of having an understanding of what linkage equilibrium and linkage disequilibrium are, and how crossing over can erode that over time, let's take a look at the next slide that includes an example of how this linkage disequilibrium can be adaptive. So let's bring this linkage disequilibrium stuff into context with natural selection. Now sometimes natural selection actually favors certain combinations of alleles, such that if those alleles are inherited together as a package into an organism, that would be more favorable, more adaptive than if those alleles were not inherited as a package, but were inherited in other combinations. So natural selection and genetic recombination can be acting in opposition to one another, because while natural selection might favor certain combinations of alleles, okay, so that's the advantage, when meiosis happens and crossing over occurs and new genetic recombinations happen, that could break up those combinations of alleles, sort of going against what natural selection favors. So selection and genetic recombination are kind of butting heads together. Now, sometimes chromosomal rearrangements, again, those happen during meiosis, unequal crossing over, can bring favorable combinations of alleles together, closer together on a chromosome, and such that they are inherited as a tight little package. And those are called supergenes. Supergenes are collections of multiple genes, all clustered and packaged tight together, that are all favoring a certain combination of characteristics. So here's an example, a great example from your textbook, with yellow monkey flowers that exist along the Pacific, the western US. And coastal populations of these monkey flowers have a certain combination of traits that are favorable for the coastal climate, which is generally having cool, moist summers, the moisture coming from fog near the ocean. So, for example, the plants are perennial. They live year-round because there's plenty of moisture and it's cool. They tend to flower late in the season because things aren't drying out. Flowers are large. They can invest in large flowers because of abundant water resources, etc. And whereas the yellow monkey flowers that are in populations inland, they have a different suite or collection of traits that are favorable for the inland climate that is hotter and drier in the summer, such that they tend to be annual, which means that their life cycle is within 1 year. They die as it gets dry and hot. They flower early in the season. It's too costly to flower later, etc. So these flowers, these plants, these populations experience very different conditions for traits such as flowering time and growth, etc. Now it turns out that the genes for those traits that I just outlined are very tightly clustered together. They form a supergene. And a chromosomal inversion, which is a type of recombination, has made coastal and inland populations unable to successfully hybridize. So basically, an accident during meiosis, the way that meiosis occurred and an inversion happened in a chromosome just by accident, ended up putting together a favorable combination of genes and chromosomes and making the order of those genes different on the coastal population chromosomes compared to the inland population chromosomes. So if you look on the left side of the figure under the coastal plant, it shows you that there's an inversion that occurred containing the genes involved in local adaptations on the coast. So the genes are ordered A2 and then D2, C2, B2 because that part of the chromosome was inverted, and so on down the chromosome. If you look on the right-hand side of the figure, the plants that live on the inland conditions, the chromosomes are depicted in purple. No inversion has happened there, and so the genes are ordered the way they were ancestrally, A, B, C, D, E, and F, all in order. So when the individuals from those two populations, coastal and inland, if they happen to hybridize close to where they come together, they can make a hybrid offspring. So they can make a hybrid individual that gets one set of chromosomes from its coastal parent and one set from its inland parent. However, that hybrid individual is going to be unable to properly do meiosis and make its own gametes. And so that basically blocks the transmission of those packages of genes from either population of moving into the other one. So yeah, if you look at those chromosomes in the hybrid individual, because the order of the genes on those two chromosomes are not in the same sequence, then that basically makes it the hybrid inviable. So in this case, a chromosomal inversion has made these two populations successfully, unable to hybridize. So therefore, accidents in meiosis ended up bringing together combinations of genes that are locally adaptive to populations in different climates. Pretty amazing and complex connection. All right, so now we're going to turn our attention to Quantitative Trait Locus Analysis, QTL analysis. This was mentioned earlier, a little bit earlier in the book. And I told you we'd come back to it. And so this is another way, this is another technique, another method that's been developed to be able to dissect complex traits and understand their genetic basis and hence their evolution. So QTL, Quantitative Trait Locus Analysis, what it does is it helps us find connections between variations in a phenotype and allelic variations. So connecting phenotype with genotype to be able to identify sources of these complex traits. So what this slide is doing is breaking down for you methodologically the steps of how a QTL is done. So step one is you take your species of interest and you inbreed it to produce basically purebred lines so that organisms become homozygous at many, many, many loci. So that's what inbreeding can do, right? Is end up, it brings together very, very close relatives. Close relatives tend to have more similar genetics. And so if you do that long enough, you're going to make highly inbred lines of organisms. So once you have these inbred lines, then you cross a male from one line with a female from the other, and you produce your F1 offspring. And of course, those F1 offspring are going to be totally heterozygous because these two lines are genetically different, inbred for different alleles. So when they mate, you're getting one allele for a gene from, male, a different allele for the same gene from, a female, and you do this all the way down, and you've got heterozygotes, pure heterozygotes, and so with lots of recombinations that occur. So then you examine the DNA of your parental lines, what you started with, and you look for DNA segments that are unique to each lineage, to each inbred lineage. You examine F2 hybrids for those very same genetic markers. You look for those markers that you found in the parent line, and you test whether the genotype at those markers is somehow associated statistically with variation in a particular phenotype that you're interested in. So there might be a locus nearby that these markers are kind of signaling. And so once you can identify these markers, then any markers that have a significant association with a phenotype, then those are what are called quantitative trait loci. Those are the loci that are associated with your quantitative trait of interest. So again, this slide is just introducing what QTL is, helps find connections between variations in phenotypes and alleles. And this slide is going through the steps of how you lead up to a QTL analysis by doing the inbreeding and the crossing and making hybrids and finding markers that are associated with those phenotypes. So here's a quick example of how a QTL analysis was able to identify the regions on the chromosomes of the monkey flower Mimulus. And so the figure that I've got here on the slide from your book is showing you where biologists have identified the genes that are responsible for these key phenotypes, complex phenotypes like throat width of the flowers, the corolla width, the tube length, etc. So how did they figure out where these genes are located on these chromosomes? How did they do that? Well, through a QTL analysis. So they followed the steps that I outlined on the previous slide. They took monkey flowers and they inbred them generation after generation to get pure inbred lines that differed in key traits. So that's step one. Then they crossed those to make F1 hybrids, and then you've got recombination. And then you cross those to get F2 hybrids, just like I outlined in the previous slide. And then they look for statistical associations between the markers that are in those F2 hybrids and the phenotype that those hybrids have. So they find those markers by looking at the parents, the original parent lines, and going, okay, well, this parental line has these unique genetic markers, and this parental line has these, genetic markers. And so that way, when they look at the F2 hybrids, they can see if they're, like I just said, a close association between having a certain genetic marker and having a phenotype. And so following that technique, they were able to, voila, identify where the regions are in the chromosomes of these plants that are responsible for these important traits, for these important phenotypes. So remember at the beginning of the chapter, the opening story was about the Oldfield mice that live along the Gulf Coast on the southeastern US, where the beach populations are much whiter than the inland populations that are brown. And the question was posed, what's the genetic basis for these complex traits that are clearly adaptive in each region? So to find out where in the genome the genes are that code for these specific traits, biologists conducted a QT analysis of the Oldfield mice. So what's step one? I put a blank there to see if you remember from a couple slides ago. I'll tell you. Step one is produce inbred lines so that you have mice that are, pure for each strain. So that's step one. So you get an inbred line of beach mice and you get an inbred line of coastal mice. So step two, using 3 mice from each population, they carried out 2 rounds of crosses. Round 1, get F1 hybrids. Round 2, get F2 hybrids. And by the time they got to the F2 generation, they had 465 progeny. So they had a bunch of mice to work with. And then by finding statistical associations between key markers from each parental line and the statistical connection between the prevalence of those markers and certain phenotypes, they're able to statistically say that this marker is related to this phenotype and this marker is related to that phenotype because the occurrence of these markers is arising much greater than chance with these particular phenotypes. That's how the process works. So they created a map of 124 markers in the Peromyscus genome. And they looked for associations between the markers and color variation. So that's step 3. And let's see what happened. So continuing with this Oldfield mice study to identify the genes that control their coat colors, after doing the F2 hybrids and getting those several hundred hybrids with different combinations of alleles from the original parents, they were able to identify 2 markers that had a very high statistical association with the coat colors being either very light or very dark in the 7 regions. Now, the 7 regions are shown on the upper part of the figure on the right side of the slide. The regions being rostrum, cheek, eyebrow, ear base, etc. And so these 2 markers, there's genes that are within those regions where the markers are, and the genes are already known to have something to do with coat color. So it was no surprise or shock that the QTL analysis ended up isolating and identifying markers that happen to contain these genes. Figure B, which is down on the bottom part of the slide, is showing you how they were able to do additional analyses and identify and pinpoint the very specific region where these markers are located, and indeed the Agouti gene ends up being the final target that's responsible for at least one of the genes, the Agouti, for the coat color. So let's keep taking a look at this. The other gene is called the MC1R gene, and so let's take a look at this some more on the next slide. Okay, so remember the QTL analysis was able to pinpoint and identify the Agouti gene and the MC1R gene as both being strongly associated with the phenotypes of the coat color through the statistical analysis. So the next step here is to bring into view what those genes do and to look at genetic variation in those genes to understand the evolution of the different coat colors on the coastal and the inland populations. So Agouti and MC1R both produce proteins that are used for synthesizing melanin, pigment, melanin. Now, one mutation that was in the MC1R gene resulted in a decrease of the MC1R's activity in terms of its receptor. And so by decreasing the activity of that MC1R receptor, that results in lighter coat color. The second mutation was in the Agouti gene, and that mutation happened to increase the expression of that gene. And the protein product of the Agouti gene interferes with the expression of MC1R, thereby decreasing coat color. So the overall bottom line is that combined, those two mutations, one that's decreasing the activity of the MC1R receptor, and the other that is increasing the expression of Agouti, those two mutations combined help to reduce melanin synthesis, resulting in lighter colored mice. So Agouti, of course, is epistatic to MC1R, and so it's got to be present for the MC1R to have a measurable effect. So these two genes are very tightly linked in terms of what they're expressing. So there we go. It's QTL analysis is extremely time intensive with all the breeding of the organisms to get inbred lines, and then doing the crossing, and then doing the statistical analyses to find associations between genetic markers and phenotypes, and going through all of this. But the result is very rewarding because we are able to pinpoint and identify those genes that are responsible for complex quantitative traits. All right, so researchers wanted to understand even more how the mutations in these two genes were controlling the expression of hair color in mainland and beach mice. So went a little bit farther, and looking at the development of embryos, they discovered that only 12 days after conception, which brings them to halfway through their entire gestation period, the melanocytes are migrating into the hairs, and this is how hairs get colored with pigment as melanocytes migrate into the hairs. And what Agouti does, that gene that suppresses the expression of the MC1R gene, Agouti delays maturation of the melanocytes. And so by delaying their maturation, they can't deposit as much or any pigment in the hairs, and that results in hairs being more of a white color. As it turns out, the expression of Agouti is strongest on the ventral side of the embryo, where the hair is light colored, and it's weakest on the dorsal side of the embryo, where the hair turns out to be the darkest. And so here it's just being pointed out that by understanding how agouti works, how it is expressed, we can tie that into the hair color or coat color of these mice to understand exactly, how they're getting the light and dark colored regions of hair on their body. It's through the differential expression of the agouti gene. So here comes the final little bit to this story, is how does agouti expression differ in beach versus mainland mice? This is what it kind of comes down to. And so the region of agouti expression spreads farther up the dorsal side of the mouse. And so you can kind of see where there's a borderline there shown with the dotted line on the mouse, where the expression is changed just enough to create light versus dark hairs on the underside versus the dorsal side of the mice. And of course, in mainland mice, the ventral hairs are dark colored, so agouti is not suppressing the expression of melanocytes down there. But in beach mice that their ventral hairs are totally white, agouti's expression is more accentuated, so suppressing the melanocytes, and they're white. So they genetically engineered mainland mice and to be homozygous for the light color allele of agouti. And by genetically engineering mainland mice to be homozygous for that allele, the boundary of the agouti expression shifted dorsally, as you might expect. So the conclusion of this is that all of this technique, all of these steps in the analysis helps us to understand the molecular basis of adaptation. We're able to apply the results of this study to the pigmentation of other animals. We're able to apply the results of this kind of study to disease, such as the role that pigment plays in skin cancer, etc. So even though this kind of a study might seem to one person like it's relatively trivial and just tells us something about mouse hair color, by extending the results of this study in other directions, then we can see that it has some pretty broad applications. Well, a newer approach since the early 2000s has enabled very detailed maps of the entire genome of species and allows us to be able to identify the markers that might be associated with particular phenotypes. So very similar to a QTL analysis, except with a genome-wide association mapping, GWA for short. Researchers do not do breeding experiments first to get inbred lines, but instead look at massive numbers, large numbers of individuals to see what their phenotypes are, and then using the mapping of the entire genome for a species. And since the early 2000s, we began to produce entire genome maps, like for human beings or the house mouse, Mus musculus, which is a model organism, etc. And so for humans, then, we have in genome-wide sequencing, and we have many, many individual humans that we can look at. And so what's done is individuals are grouped by phenotype. So, okay, all of these individuals have, for example, coronary heart disease. And all these individuals we're grouping together have Crohn's disease, and these have rheumatoid arthritis, etc. And then looking marker for marker across the entire genome, biologists identify alleles that are correlated with their trait of interest. So they can find statistical associations between a particular disease and then the frequency of certain markers that those people have compared to other people that keep popping up over and over and over again. And so by identifying through the entire genome which markers seem to pop up statistically and be associated with certain diseases, for example, scientists, biologists have been able to find the genes that are correlated with diseases like the ones shown on this page. So this is a very powerful approach that has a lot of promise for not only being applicable to humans, but of course other species, for being able to track down those genes that are associated with particular phenotypes.